I am going to tell this plainly, without the softening language my department preferred at the time. The softening language was how we avoided responsibility for what we did not understand, and I am done with that. My name is Sareth Venn. For 11 years I held the title of Senior Containment Virologist aboard Prison Station Vethara 6, a Commonwealth detention facility positioned at the edge of the Vareth Transit Corridor. Roughly 40 light minutes from the nearest inhabited body, processed detainees from 17 species. We ran controlled biological assessments as a matter of standard intake protocol. None of what happened with the human was outside the bounds of what our department considered routine. That is the part I have spent 4 years trying to reconcile. The human arrived on a transport transfer in the 3rd week of the Vethura quarter cycle. His name, as rendered in our intake system from his own identification documents, was Marcus Doyle. His file listed him as a civilian freight coordinator, detained pending an investigation into unlicensed passage through a Commonwealth exclusion corridor. Not a soldier, not a combatant. A middle-aged man with calloused hands and a low-grade annoyance at the delays that had accumulated in his paperwork over the preceding 6 months. He was placed in Containment Section 4, which we used for species whose baseline biology was still being catalogued. Humans were relatively new to the Commonwealth Detention Registry. We had processed 3 before him. The intake assessments on those 3 had been unremarkable, or so we believed at the time, though I would later revise that conclusion substantially. The decision to include Doyle in the Vareth 9 pathogen study was mine. I want to be exact about that. The study was examining a Class IV containment agent, a synthetic organism our department had developed specifically, use in sealed environments, capable of incapacitating detainees who breach containment without requiring direct guard intervention. It was aerosolized, fast-replicating in most known respiratory biologies, and self-limiting, designed to degrade within 72 hours, leaving no lasting systemic damage in the host. We had tested it across 11 species. The success rate, meaning the rate at which it achieved full incapacitation within the target window, was 100%. The study protocol called for a passive control, a 12th subject who would receive an identical exposure to the pathogen, but in whom we expected no significant response, a biological baseline, essentially flat, against which we could measure the active results. I selected Doyle because human respiratory biology was the least characterized in our registry. We expected nothing. That was the precise, documented, stated reason. We expected nothing from him. I introduced the Vareth 9 agent into Section 4's ventilation on the 41st hour of his detention. Standard concentration, standard delivery window. I returned to the monitoring suite and waited. The first anomaly appeared at hour 4 post-exposure. In every prior trial, the Vareth 9 colony count within a subject's respiratory tissue showed exponential growth beginning at hour 2 and reaching peak saturation by hour 6. We had a very clean curve. I knew it by memory. What I saw on Doyle's bioscanner was not that curve. The colony count had risen modestly through hour 2, plateaued at hour 3, and by hour 4 had leveled in a way I initially attributed to a delivery error. I ran a diagnostic on the ventilation feed. The feed was clean. By hour 6, the colony count had begun to fall. I called Warden Thessa and 2 of her staff to the monitoring suite. I did not tell them what I suspected because I did not yet know what to suspect. I showed them the readout. Thessa asked if the pathogen had degraded prematurely. I told her the degradation cycle was not due for another 60 hours. She looked at the numbers for a long time, without saying anything. On the internal cell feed, Doyle was sitting on his bunk eating from a meal ration he had requested, about an hour after exposure. He had flagged a mild headache and a low fever through the intercom. 38.2 degrees by the cell's surface thermometer, which was elevated but not dramatically so. He asked how much longer the paperwork delay was going to take. I noted this in the log. A subject receiving a full saturation dose of Vareth 9 at hour 6 should have been fully incapacitated. Doyle was mildly irritable and hungry. By hour 12, I had pulled every comparable dataset I could access on human immune system function. What I found there I will attempt to describe accurately, because I do not think my department had grasped it before this moment, and I am not certain the broader Commonwealth Registry had either. Human beings have been host to microorganisms, bacteria, viruses, parasites, fungi, and entities that do not map cleanly onto any of those categories for approximately 4 billion years. That number is not a metaphor. Their evolutionary history is, in a meaningful biological sense, an unbroken arms race between their immune architecture and a vast, constantly mutated population of organisms that have been trying to kill them since before their planet had stable land masses. The human immune system is not a simple barrier. It is a layered, adaptive, memory-capable system that identifies novel pathogens, sequences their surface proteins, and begins producing targeted responses within hours of first contact. It does not wait to be overwhelmed. Catalogues. It responds. It remembers. The Verith-9 agent had never encountered anything like it. I watched the bioscanner readouts through the night cycle. The colony count dropped below 50% at hour 14, below 20% at hour 18. At hour 22, the scanner was having difficulty locating sufficient sample mass to generate a reliable count. The pathogen, which had never failed to fully saturate a host organism in 11 trials across 11 species, was being dismantled. Not passively. There was nothing passive about the immune cascade I was watching. Doyle's body was hunting it. He slept for about 6 hours in the middle of this. He snored. When he woke, he flagged a mild sore throat and asked again about his paperwork. I noted that his fever had dropped to 37.6, essentially baseline for the species. He ate another ration. He did a series of stretching exercises near his bunk that I do not think he was aware we were recording. There was no indication on his face or in his behavior that he understood anything unusual was happening inside him. As far as he was concerned, he had a mild cold. He was mildly inconvenienced. At hour 26, bus scanner returned a colony count of 0. I ran the confirmation protocol 3 times. Each time, 0. I stood in the monitoring suite with Thessa and her 2 wardens for a while without speaking. One of the wardens asked if the agent had self-terminated early due to a batch defect. I did not answer immediately because I was looking at the other data on the screen. The immune marker panel, which was still elevated, still active. The cascade still running, Doyle's body still searching for something that was no longer there. Searching with the systematic patience of a biology that had never once in 4 billion years been given a reason to stand down quickly. I told the warden that the batch was not defective. I told her the degradation curve was not a technical anomaly. I told her what she was looking at was the result of a pathogen entering a host organism whose immune system had been shaped by a longer and more relentless evolutionary conflict than anything we had modeled. She asked what that meant for the study. I said I would need to file a revised protocol report. I did not immediately say what I was thinking, was that the Verith-9 agent, every version of it, every concentration, every delivery method, would likely need to be reclassified as ineffective against human subjects. I did not immediately say that the 11 successful trials we had logged now felt like 11 experiments we had never thought to run correctly. I did not say that the word passive, as applied to a human control subject, was a category error that I should have caught before I ever opened that ventilation feed. I filed the report the following morning. 4 pages of data, 2 pages of analysis, 1 page of methodology notes. On the final line of the document, under the heading Operational Advisory, I wrote the following sentence and flagged it for distribution to every detention facility in the Commonwealth registry. Human subjects are not to be used as biological controls in pathogen studies. Their immune response does not establish a baseline. It establishes a ceiling. Doyle was processed and released 11 days later when his paperwork cleared. He signed his discharge documents, collected his personal items, and walked out of Section 4 without looking back. He did not know what had happened in his cell. There was no reason to tell him. From his perspective, he had caught a minor illness, waited out a bureaucratic delay, and gone home. I have thought about that often since. The thing that unmade 11 months of pathogen research and rewrote Commonwealth detention protocol was, to the man it happened inside, nothing more than a head cold. I think about what it means that he never noticed.